So this is a high-dimensional workflow, FlowSOM T-Rex, and ClusterExplorer. Just to give you an outline of what we're going to discuss today, it's those three things: FlowSOM, T-Rex, and ClusterExplorer. I think T-Rex is supposed to be all caps. Actually, I did spell that wrong. Um, FlowSOM is for making meta clusters or clusters in general. It's a clustering plugin, third-party plugin available in Flojo. Uh, T-Rex is for condition comparison. So it's a really cool algorithm for looking at two different two different states and comparing those two different states to see where the differences are between those two different conditions. Um, Again, it's a third-party algorithm that we will look more at in a little bit. And Cluster Explorer is a built-in tool in Flojo that we can use for phenotyping. It makes heat maps, line charts, etc. And so this is just, again, a brief outline of what we're going to discuss today. I'm going to move through these slides pretty fast, but if I blaze through something in the slides, don't worry, I'm going to do a live demonstration after the slides so we can see it in action. So I'm going to go through the slides kind of quick just to get to that live demo. So why do we want to do clustering in the first place? So when we're thinking about high parameter analysis, like a lot of assays these days often have 20 plus stains. And so it's a lot of data to parse. And so we could do it the old-fashioned way, which is drawing a bunch of gates, and that's what's shown here. Gates are great, and I do think they're important, and probably I would still do gates in almost every analysis. And they can get us to these results, but it's a lot of work, and sometimes where we set our gates is a little ambiguous. You can see for a lot of these populations here, like the divide between positive and negative isn't super clear. And so we can use clustering methods as like a validation tool or verification tool or another tool to generate subpopulations of interest and group our data together into its phenotypes, but just using machine learning instead of using manual gates on 2D plots. Okay, so that's what this high parameter analysis clustering tools are. These three photos represent three clustering tools available in Flojo 10. We have Phenograph, XShift, and FlowSum. I should have mentioned at the top of this webinar, this today will be covering basically everything will be in Flojo 10. There is a new version available on our website, Flojo version 11. The first release doesn't have all these algorithms incorporated into it yet. It just has XShift available in Flojo 11. And in general, in general, Flojo 11 is great for new users. But for any user who wants to do like really advanced stuff, I would still stay in Flojo 10 for right now because we're still working on adding some of those new or some of those current uh, platforms into Flojo 11. Okay, so today we're going to be focusing on FlowSum, which is the far right image here. Um, the basic goals of a high parameter analysis, typically I would start by defining the populations using clustering. That's step one on the far left here, using one of the clustering algorithms I just mentioned. Once we've defined our clusters using a clustering algorithm, we'll have to visualize and identify the phenotypes of those clusters. So that's the second step here. When we do our gates, when we get to our final gated population, we know what the phenotype is because we've defined it through the series of gates. With clustering, the algorithm will return results, but it's not necessarily going to label what the different groupings are that it's returning. And so we're going to have to use some sort of visualization technique or cluster explorer to identify and call the phenotypes and say what the clusters represent. And then we can also compare how the clusters are changing between different conditions. And so that's kind of what T-Rex is going to be doing for us, is it's going to compare two different conditions and say, hey, like what is the difference? Like what's changing between these two conditions and figure that out for us. So we're gonna keep moving these slides. I see a few new people have joined us. Um, Thanks for joining. We're talking about clustering high dimensional analysis today with FlowSum, T-Rex, and Cluster Explorer. We're gonna do some slides quickly and then go to a live demonstration. So in a high dimensional workflow, we typically have these 9 steps or so. Um, Normalization, data QZ, that's like the first thing we might do just to clean up our data and get down to like our good live events that we want to look at for future analysis. And so I've already done those 2 steps before my live demo today. So most of us probably are familiar with QC and gating and cleaning up to our live cells. Um, after we get down to our live cells, we might do some annotation to identify, like anti-R samples to identify which samples are from the different conditions. Um, and then we'll group the data together using concatenation. So concatenation just combines the data together. And FlowJet 10, the concatenation process, will make a new FCS file. So we'll choose populations to combine together, and then we'll create a new FCS file. In FlowJet version 11, there's actually going to be this new cool feature called virtual concatenation. Virtual concatenation lets us pool all the data together at any time, and so it's a really fast way to concatenate. It's going to be nice going forward, but again, I'm not using FlowJet version 11 today just because FlowSOM and T-Rex aren't in FlowJet version 11 yet, and so um, we're going to stay in FlowJet version 10 for now, but someday in the future, it's going to be a much faster process, I think. Dimensionality reduction. So dimension reduction is the ability to take these multiple parameters, 10, 20, 30 parameters, however many you were working with, and collapse it down to two dimensions into a two-dimensional plot that to separate the data based on similarities. And so it's like a type of machine learning. It doesn't do clustering per se. It's really just kind of grouping the data together visually, but it's going to be an important step of our process. And then really comes to the three that we're focused on today, which are these three, which is clustering, phenotyping, and comparison. So clustering, again, that's going to be what we're using FlowSOM for today, just grouping the data together into populations, representing hopefully the different phenotypes. And then once we have our clusters, we're going to do phenotyping, so identifying what those populations represent and labeling them ideally, based on what we believe those populations are. And so phenotyping, again, we'll be doing probably by using ClusterExplorer, uh, and so identifying the different marker expression for our populations and then labeling them based on marker expression. And the last step we're going to do today is comparison. And so using T-Rex, we're going to compare our two different conditions. And so we can compare like a healthy to disease state, or we can compare untreated to a treated state and see where the differences are between those two conditions, what cell populations are changing, and identify those cell populations. Isolation, that ninth step there, that's the concept that, once we've done all this analysis and identified an interesting population, we could isolate it and we could even send it back to the sorter. If we're using like a S8, we could feed that data back into the S8 sorter and then sort that population and isolate and look at those cells that we've identified through this process. But that's beyond what we're doing today. Okay, so I'm blazing through these slides again. If you do have questions, though, feel free to email me. Our email is flojo@bd.com. Or if you have questions, feel free to drop them in the chat right now, the chat or the Q&A, and I'll try to answer them as we're going along. Um, I just wanted to briefly demonstrate dimensional reduction. I talked about it briefly earlier, but again, the goal of dimensional reduction is just to represent high dimensional data in low dimensional space. So we're taking many parameters and describing them in a 2D plot like you can see here, and grouping the cells based on their similarities of those parameters. So the plot on the right is a t-SNE plot. They're pretty common, just visual representations of the data. It's going to be similar to clustering. We would expect our clusters to be split up similar to the islands that we see here in this t-SNE plot. But the visual representation we see here um, is mostly for visual purposes. Okay, whoops. So again, the goal with the t-SNE plot is to separate the data and visualize it based on its different phenotypes. And so you can see here, this t-SNE did a pretty good job of separating my different phenotypes. You can see my B cells, monocytes, CD4, CD8, and NK cells were all pretty well separated. It split some of these up further. For instance, my CD8 cells, it split up a few different ways, probably based on like activation state. And so there's different, within each phenotype, there's even more different subtypes that could be expressed. And so the machine learning sometimes can group the big phenotypes, but sometimes it'll also split them up based on those changes of like naive versus memory or other substates. So dimensional reduction is a good visualization tool. We have a lot of different dimensional reduction options available in FlowJo. 8 are pictured on the right there. t-SNE is built into FlowJo. It's the easiest and pretty common. UMAP is also pretty common. There's some less common algorithms we also have available as plugins that you can try out on our website. Um, Again, the plugins are available on if you go to flowjo.com and go to like downloads, it should say plugins. Or if you search Flowjo Exchange, you should find the plugins available there, and that can get you access to everything on this list besides tsne is available as a plugin. Okay. So another visualization tool that we'll be using today is multigraph overlays. This is built into Flowjo. You can just right click on any plot with a single population in the layout, and you can say multigraph overlays, and it will make a visual similar to what you see on the right here, where it has a heat map parameter. So we're looking at that same tsne we were looking at before, but with a heat map parameter now, and so it's highlighting, the expression of these different markers. And so we can see what populations contain each marker. So for instance, granzyme B, it looks like these two populations have expression of granzyme B. Um, CD8 is highly expressed down here, it looks like, so those are probably our CD8 cells. And so you can get an idea quickly of, what, and this is our CD4 up here, it looks like. So you can quickly get an idea of what these different islands represent just using multigraph overlays. So I think multigraph overlays is a really simple way to quickly visualize data. And again, it's just a right click option when you're in the layouts in Flowjo, and you can choose multigraph overlays. And this is multigraph color mapping, and it's a great visualization tool. Um, okay. I'm going to dive into the clustering now. So the clustering is the meat and potatoes of what we're talking about today. Clustering allows us to define populations automatically without having to do individual gating down to our populations of interest. Um, it uses, computational algorithms. It uses the data, the parameters, and so it can be unbiased. It can help us quickly get to final results. Some of the downsides of clustering is once it generates those results, we have to, usually it'll just call them like cluster one, cluster two, cluster three. You can actually kind of see that here on the right. We have our manual gates up here at the top. And you can see we've done the gating ourselves, so we should know what those populations represent. But if we just do clustering, we'll get the results really fast, but it's just going to label it one, two, three. It doesn't necessarily know what those populations are, and so we have to further interrogate to find out what those populations, like what phenotype is actually represented by those populations. It just knows these cells are similar, so it's grouping them together. So we'll have to identify the phenotypes. Clustering also is unpredictable. That's one of the benefits, but it could also be a con because maybe when we're doing FlowSOM, we're trying to specify a certain number of clusters to get a certain number of phenotypes. So maybe I just want my major subsets, but it might end up splitting one of my major subsets. So it might end up splitting my CD8 cells into two different populations based off naive and memory, and that might not be what I want every case. It might also make too many clusters or too few. I mean, we can't control, since this unbiased method, um, what kind of results we're going to get. We also, with clustering, you don't get the final gates, and so you can't really go back and adjust the results by just tweaking some gates. The results you get are kind of the results you get, and the only way to get new results would be to run the clustering again with different initial settings. Okay, again, I'm blazing along, but if anyone has questions, feel free to throw them in the Q&A or chat at any time, and I'm happy to answer them. So the clustering we're going to be doing today is FlowSOM. FlowSOM uses self-organizing maps and k-nearest neighbors to group the data together. Self-organizing maps is just an iterative process that kind of just groups similar data together. FlowSOM specifically, by default, with our default settings, we'll be making 100 subpopulations, like tiny clusters basically, that it then groups in together to generate however many clusters we specify. So a big advantage of FlowSOM is it's a clustering algorithm where you can specify a number of meta clusters for it to return. And so whatever number of clusters we tell it to give us, that's the number it's going to generate. So if I tell cluster to generate 10 clusters, it's going to give me a result with 10 clusters. It's going to do that by making first many, many smaller clusters and then grouping them together into those 10 that I've defined. FlowSOM's pretty fast, which is a big advantage. It also produces some additional visuals, like here on the right, it makes these minimum spanning trees and heat maps that folks like. So it makes, it has each of the clusters represented here in the rows or with the colors in the minimum spanning tree. And you can see each of the parameters as the columns. And so there's some nice visuals for FlowSOM that makes by default. A big advantage of FlowSOM 2 that we aren't really going to look at during our live demo today is that FlowSOM can use this thing called apply a map. And so it allows you to apply the FlowSOM results from one run onto a future run. And so this is useful if you expect your data to have the same phenotypes across the different runs and you're doing like a longitudinal study. You can run FlowSOM once on the initial run and then save its results. And then at a later time, you can run FlowSOM again using the initial run's map, which is the old results, and it will generate the same phenotypic populations, but just with different percentages to represent the change that's been, happened between the two different time points. And so the apply map tool is a really useful tool, but again, we're not going to necessarily be looking at it today because I already have all my data. There's also some downsides to the apply map tool. It only works well if all of the populations are well represented in what you originally run FlowSum on. So you can't apply a map onto a future run and expect it to get new populations. It only is going to find the populations that's already found in the first run. So again, if you want to learn more about apply a map or any of FlowSum's features, you can watch, there's a FlowSum video on our website that's going to go into much more detail on just FlowSum. You can also read the original paper, um, which is linked there in the bottom right-hand corner of this slide, and you can also find it linked on the FlowSum plugin page on our website. Um, okay, so I guess I didn't mention the challenges. One downside of FlowSum is that you have to specify a specific number of meta clusters. So oftentimes you might not know how many clusters you want, especially if you're looking for like new or novel populations. Um, and so with FlowSum, you do have to guess a number of clusters, or you'd have to like iterate through a number of clusters and run the algorithm multiple times, or you'd have to somehow otherwise determine the number of clusters, but you have to give a number of clusters. You can't, the other algorithms like phenograph and XShift, they'll generate a number of clusters based on what the algorithm thinks is best, but for FlowSum, you have to specify a number. Okay, if there's any questions, again, feel free to put them in the chat, but I'm going to keep moving forward. Um, The other clustering that we're going to be looking at today is T-Rex. Really, T-Rex is going to be used more for comparing two conditions. And so it's a really neat tool for comparing uh, like two conditions. And the way it does that is it creates a dimensionally reduced plot, either a TC or UMAP. I'm going to be using UMAP today. And so it, creates a plot like the ones we see on the right, and then it identifies areas that are different between the two conditions in this plot space. And so it uses dbScan to group the data together based on the differences it sees between the two different plots for the two different data sets. And it will cluster the data based on those differences. And so you'll get clusters similar to these ones. And you can see the label on the bottom right-hand corner. It shows that the clusters that are blue, 95% of the cells are from this ACLL condition. Um, and so, these areas of the plot only represent, the cells in these areas only represent this condition. The red cells are only from the other condition, the CLL condition. And then the areas that are kind of more gray, those areas are shared between the two conditions. And so you can see um, which populations belong to each of the two conditions. Okay, so yeah, it also will cluster the data. It'll cluster these things together so that way we can, identify them and look at them further. Okay, and last but not least, phenotyping. So if we actually want to understand the identity of these populations, again, we'll want to use something like ClusterExplorer to see the different expression of the markers within those populations. And so you can see in ClusterExplorer here, I have my different clusters listed on the left, and it's showing me this heat map, giving me the different marker expression for those clusters. ClusterExplorer makes a few visuals, makes heat maps, line charts, bar charts, and interactive plots that we can use to explore the data. ClusterExplorer is built in now, so if you had before, it used to be a standalone plugin, but in the newest versions, it's built in. So if you have a plugin version, you want to delete it and just use the built-in new version because it's going to be better. Here's another view of ClusterExplorer. Again, it's a way of visualizing data. We can use it to see marker expression, make line plots, all kinds of comparisons of those clusters to try and identify what they are. But I think it'll be easier to explain once we go to the live demo, which is what I want to do now. Okay, so If you want to download the plugins that I'm going to demonstrate today, you can do that. It's www.flowjo.com/exchange, this first link here. That'll find the plugins. You have to download them. FlowSOM and T-Rex both require R, since the person who developed these algorithms developed them in R. So that'll require download and setup of the R algorithm. Uh, you can find information about how to set up plugins or anything else, or read more about the plugins themselves, in our documentation, which is just docs.flowjo.com. Um, you can also find videos. I think there's videos on some of these specific plugins on our website, Flowjo University, and so that's a great place for resources. You can also email me. I didn't put it here, but you can always email us at flowjo@bd.com, and we're always happy to answer more questions. But now I'm going to leave the PowerPoint part of my demo, and let's dive into actually doing the analysis. So I guess I don't have it open right now, but let's go ahead and open up this advanced analysis thing here, and let's open up the one that says start, which is this one. Okay. So to start my analysis, I've done some gating. So I've already done some cleanup gates. I expect most people on this webinar might be familiar with just doing like a size gate, a single cells gate, and a live gate to get down to my live cells. I've done some additional gating, but I'm not going to worry about that today. Um, we're just going to use those live cells and kind of go from there for the rest of our analysis. So this data set, oh, I should mention the data set comes from Flow repository. It is from Marie Frutoso. It is a data set comparing disease and healthy patients with respiratory diseases. Specifically, I'll be looking at COVID-19 today. Um, you can find it if you put in this repository ID to Flow repository. I've found that Flow repository has been working great recently, but you can give it a shot if you want to try and download this data. You can download it here. It's a great data set. Appreciate the author for making it available for us to use in open source fashion. Um, and so I'm going to go ahead and dive in. I've split the data up into 4 samples in the healthy condition and 4 from the disease. I chose to do an even number of samples from each just so it's more comparable. T-Rex will only compare an even number of cells from each of the 2 conditions, so might as well just make things even from the start. And so on the topic of making things even from the start, you might imagine that in a lot of cases, the number of cells acquired in each of the samples might be different. Like you can see, this one only has closer to 900,000, whereas one of the ones above have closer to 1,200,000. And so, or sorry, that's actually 1,200,000, I think. Um, and so, anyways, there's some differences in just the cell counts from sample to sample. And so this one actually has a lot less here. You can see it only has like 500,000. And so one thing I might do at first is just to make things more comparable between my 8 different samples here, is I'm just going to want to go ahead and downsample these populations. And so downsample is a plugin. It's available under Workspace plugins. Downsample, again, if you haven't downloaded it and set it up, you'll have to do so from our website. If you go to Flowjo Exchange, you can find the downsample plugin and download it and put it in your plugins folder, and it should be available. The downsample plugin, though, lets us specify a number of cells. So if I choose downsample here, it'll open up this little window where I can specify a number of cells, like 200,000. Actually, that's way too many. Let's do 50,000. I can specify a number of cells to subset the population from, so it's going to take an even distribution of the cells within my live cell population and subset it to get it down to exactly 50,000 cells. So that way I can have the same number of cells, 50,000, from each of my 8 samples. Okay, so I'm going to select the downsample node and downsample population. Once I've run it on this first sample here, I can just right click, copy analysis to group to apply the same operation to all my samples. This notification pops up. You can ignore it. It's just saying that because it tried to apply the downsample population before it finished calculating, but in the time I've been talking, it's already super done calculating. And so you can see it has this downsample population subset of 50,000 cells from each of the samples in my group here. I actually meant to name it something different, but that's fine. Okay, so I just call it, by default, it'll default to downsample DP. I had the option to name it back here, but I didn't take it. So it just has a generic name. That's fine though, I can keep trucking. This downsample node it's a great idea to apply again both the downsample node and the downsample population to the entire group. You'll have to apply this down arrow node to the group to actually do the downsample calculation for each of the samples. But I want to also apply this downsample population to the group just that way I can have it there for any group-owned gates I might be applying to this downsample population. But once I'm finished downsampling all my samples, I can delete the downsample arrow from the group because I don't really need it, and just leave this downsample population because that's the part that actually has the cells that I care about. So again, the reason I downsampled all my populations was just to get the same number of cells contributed from each of my populations. Now I'm going to go ahead and combine my populations, my different samples together. So I want to concatenate the data to combine it together so I can do machine learning on the combined data, because most machine learning works best if I hand the machine learning algorithm all a representative subset of all the cells. So that way it's able to, do its machine learning analysis on all the different conditions in cells and populations and generate results that way. Again, FlowSOM has an apply on map feature. We have an embed feature that also can be used to apply the results from one machine learning result onto another sample. But it's going to work better if I give it all the information initially and it can run its initial machine learning on a representative subset of all the samples. Um, because if it generates results and then I try to apply those results to another sample later, it won't make anything new occur. It's just going to use this, it's going to fit the old results. Um, okay, so let's go ahead and combine this data together. Before I combine it together though, let's add some annotation to it because annotating the data is going to be really important for understanding what's what later. I'm going to annotate it by adding some keywords. The keyword button is under the workspace tab here. You can choose keywords, add keyword. Specimen ID is a great keyword. Let's add it. I'm going to add a second keyword. I'm going to call this one state for my different disease states, either healthy or diseased or COVID. For these keywords, I'm going to add some values. For the 8 samples I have in this experiment, I'm just going to add a keyword value that says 1 through 8. A quick way to add keyword values is to go back to the keywords button, say create keyword value series. Since I selected the specimen column, it's going to choose that by default here, but you could always click and choose what keyword to add the value to. And it's just going to do 1 through 8 by default, so let's go ahead and press OK so it can do that. And so it's just labeling my samples 1 through 8. So if I wanted to know what samples what later, I can just use this ID. In addition to that, I'm going to go to each of my 2 different states and label the healthy cells 1, so like a state of 1, and my disease cells will have 2. I can also like type in 2 and then select it and say copy value to group, and that's a fast way to, if I had many, many samples, that'd be a fast way to apply this value to all of them. Okay. So let's go ahead and combine my data together. I will choose my downsample population, right click, and say select equivalent nodes. And so that select equivalent nodes will choose the same downsample population for each of my samples in this disease data set. So I have all 8 of them chosen. I can right click, export / concatenate this data, choose concatenate. I think I go to advanced options. So there's a bunch of options here in the concatenation step. First, I want to choose a destination. I will choose my desktop. Mac will ask me if I want to let it. I will say yes. So I'll choose the desktop. Then down here we have a few options. We can actually do downsampling built in here if I do the include no more than option. I don't like the include no more than option though, just because I like to be able to go back and reference this population later. So having it there available as a population is kind of nice. For parameters, I'm going to choose a custom set of parameter and choose view / edit. because it'll try to include my downsample parameter by default, and I don't need that downsample parameter. It doesn't do anything after it creates the downsample population, so I don't want it. So that's perfect. I just want all the parameters. We choose the uncompensated parameters typically just because we can apply a compensation matrix to this data later. If the data had vastly different compensation matrices between the different samples, we would choose the compensated parameters, and then instead of writing the raw data into the file, it'll just write the compensated data, but then we couldn't apply a compensation matrix again to that same data. So choosing the uncompensated parameters will work well here, though. I'll press OK. I can give it a name. I might like call it something like combined live for like combining the live cells. And then down here where it says additional parameters, I want to choose this, and I just want to choose those parameters I made, so the specimen ID and the state parameters. I want to include that with the concatenation, and when I include it here, it's going to add them as new parameters available in the concatenated file, so I can separate them based on these parameters later. I can divide the data. I like to untick this spread distribution of keyword tick box, just because I don't want that artificial spread. I don't think it looks nice. Um, if I press concatenate, it's going to generate my concatenated file there on the desktop. I can load that concatenated file back into FlowJo. I might make a group for it. So let's go ahead and make a combined group. So this group will represent my combined data. So all 8 of my files combined together. I can just drag and drop it to load it. You can see it has the compensation applied because it just uses the compensation from those original 8 files. None of the gates are applied yet, but if I wanted to, I could go here, copy the gates. I don't want to copy that down sample population because it wouldn't make sense. But I can copy the rest of these gates and just drag and drop them onto my combined group here. You'll notice it says 100% for the first few gates because I only selected my live cells for the combined file. So that makes sense that all 100% of the cells are in the size, single cells, and live gates. And so this is where I'd probably start with my machine learning going forward. Just to show some other things, I will double click on my live population. If you remember, we gave it those additional keywords when we were concatenating. We can see those keywords here. So here's the specimen ID keyword. I'm just making a histogram of it. It has 8 peaks. That represents the 8 different samples, 50,000 cells each, that was used to create the original concatenated file. More importantly, though, is that state condition, which has 2 peaks, one for healthy, one for disease, based on the keywords I assigned. So if I right click on this, I can say create gates on peaks. It will automatically gate these 2 peaks, and you can see it returns these 2 populations here. I can right click and rename these, and I know that this first one is my healthy because it has a value of 1. And if you recall, I assigned a value of 1 manually to my healthy state. So if I go to my healthy state, you can see it has a value of 1 for all of these. And then my disease state is this one that has a value of 2 for all of these. So I can go ahead and rename the disease population, this state pop2, I can rename it to COVID because that's what this represents. So the reason I did that is because it'll be useful later when we're comparing the 2 different conditions. But for now, the first thing I want to do is do my machine learning. So I could just dive right into FlowSOM and run it here. But again, one of the downsides of FlowSOM is I have to specify a number of clusters that FlowSOM will return. And so I could guess at the number of clusters I think are in this data set, just based on what markers I have available. But I think a good first step would be to run dimensionality reduction, which I probably was going to run anyways. And so I might start by running tsne. Tsne is built in. It's a button just here under the workspace tab. When I press tsne, it opens up this window. For all of our machine learning, we typically want to run giving it any parameters that we haven't used yet in the analysis. So in this case, for instance, I've already used my live dead stain, so I wouldn't give it that. But all the other parameters I haven't used yet because we still have, CD3 subsets, B cell subsets, monocyte subsets available under my live cell population. There's a few different things we can fiddle with here in tsne to change the results, but in general, the defaults are pretty good. The only thing you might want to adjust occasionally is the perplexity, which will affect the way the data is grouped together. So if you don't like the visual results of t-SNE, you could try modifying the perplexity. Perplexity values between 15 and 120 are typically a pretty good range. 30 is a good starting place. And so I'm just gonna, I just run it with the default personally. I wouldn't really adjust the learning rate or iterations necessarily. We're already doing opsne, so the iterations will be cut off short if it converges on a result. Um, and again, this is just going to generate a visual plot for us, a two-dimensional plot. When I press run, I'm not going to press run because it'll take time. And so instead, I'll use the magic of preparing ahead of time to show you the results. So let's pretend I press run, and let's pretend we wait 5 minutes for it to get results. Um, we will get results that look like this. It's this second workspace where I've already saved the t-SNE results. And so I've already ran the machine learning and generated the second workspace just so we don't have to wait. I ran both the t-SNE. I also ran FlowSOM, so I will show FlowSOM, but then I already have the results secretly done here, so we don't have to wait for them. Um, okay, so when t-SNE finishes running, it will return results, and the results will look like this. So that's the same settings, the same data that we were just looking at. I think it's you can see it's the exact same number of cells. It's the same data. And so this is the results that it would have returned if I had press run. And we can view these in the layout editor. If I just right click on this, I can say copy to the layout editor, and that will put it here. If the layout editor is not already open, you'll have to right click and copy to the layout editor twice, once to open it and once to copy it in. So I'm copying it to the layout editor so we can view the data later and manipulate it. But we can look at the graph window maybe for a second here. Just look at the results. You can see it's grouped my data together based off the marker expression, and hopefully it's grouped it into our different phenotypes of interest. You can see I have a few different phenotypes, and we can maybe just count the number of islands we see here as a good starting place for getting an idea of the number of clusters we might expect FlowSOM to return. So, for instance, I have 1, 2, 3, 4 big islands, 5 big islands, 6 maybe big islands. So I'm going to say I have 6 big islands, but I have some smaller islands like 7, 8, 9, 10, 11-ish, maybe 12. Um, so, somewhere between 6 and 12 is the number I probably want to specify. If I specify 6, I know for sure it's going to group a lot of these smaller islands together, or potentially with some of my bigger islands. I don't necessarily want that. But if I go too much beyond 12, what it's going to start to do is it's going to start to break some of my big islands in half, because you can see like within these big islands, there's still some variation um, and some splits, and those splits probably represent, like for instance, this island might represent my CD8 cells, and within it we might see like CD45RA positive and CD45RA negative, or basically naive versus memory subsets that are being split up. And so as I specify a higher and higher number, it might start to split up subsets within my major phenotypes, and I don't necessarily want it to do that on this first pass. And so there's a tricky balance here of trying to choose a number of clusters that will identify all my major phenotypes without further subsetting those phenotypes into smaller populations that I'm not as interested yet in this step. So in this case, I think 11 is going to be a pretty good number for us. I think 11 is going to grab all the islands without splitting anything up too much. And I've already run FlowSOM a few times here, so I know 11 is going to be a good number that works. But um, what I did personally to arrive at the number of 11 is you could run FlowSOM with 10 clusters, it'll return results. Run it with 11, it'll return results. Run it with 12, and then just kind of compare the differences and see which one you think represents your data the best. Okay. So let's go ahead and run FlowSOM. To run FlowSOM, I'm going to choose my live cell population again. I'm going to go up to my workspace tab, go to plugins, choose the FlowSOM plugin. It'll open this interface here. Again, I'm just going to choose all my compensated parameters that I haven't used yet. And so again, I'm going to exclude live dead, but everything else should be appropriate since I'm just trying to identify all my major subsets. Specify the number of clusters. This is the most important step about flows, and that was this part I was just considering, is how many clusters I want to specify. Again, in this case, eyeballing it, I see 11 islands. 11 should be a pretty good number of clusters. Um, you could go up and down and try and see if it represents your data better, but I kind of already know I want to do 11. Um, there's a few other things you can tweak in the FlowSOM algorithm, the grid size, um, and then some different options for visualizations that you can generate. I'll let you read the FlowSOM documentation to understand these options more. There's really nothing I would change. The defaults are pretty good. The only default I would change, sorry, is the number of meta clusters and choosing the correct parameters. But otherwise, you don't need to change the defaults. Um, if you do want to learn more about any of these options, though, I would check out the FlowSOM documentation. And again, there's even this cool apply on map feature if you want to transfer results from one FlowSOM analysis to another. Okay, so normally I'd press OK and it would run FlowSOM and calculate and generate results for us. FlowSOM's pretty fast, but what's even faster is doing the results ahead of time. So let's pretend I press OK and it runs the algorithm and it's going to generate the results. Luckily, I've already done this, and so the results are here. So when FlowSOM finishes, it's going to return this. It's going to return this a/b derived parameter. TC does the same thing. Just keep these parameters. These parameters represent the results. But FlowSOM's also going to generate a bunch of gates. And so these are the gates that FlowSOM generates here in our workspace. It makes these different populations. So it made the number of populations I specified, which is 11. You can see these populations represented here. FlowSOM also makes a lot of outputs. I can find these outputs. It makes a folder next to the workspace with the same name as the workspace. Inside this folder, there are subfolders. Inside the FlowSOM subfolder, there's this live folder with the FlowSOM outputs. And so there's a lot of different outputs FlowSOM makes that you could dive into. People, for instance, really like the minimum spanning trees that FlowSOM makes. Um, it's great. These results all come from the FlowSOM R algorithm, and you could check them out. But for today, we're really going to focus more on the clusters that FlowSOM makes because that's really the meat and potatoes of what we're trying to get from FlowSOM. So FlowSOM makes these clusters, and we can evaluate the clusters by just selecting them all. I'm just on my keyboard holding down the Shift key and then clicking on the bottommost cluster to highlight all the clusters, and then just dragging and dropping them into my t-SNE plot here in the layout to overlay them. So I just dragged and dropped them on top of this t-SNE to visualize these clusters. And you can see it matches the t-SNE results pretty well. There's maybe some disagreement, but um, the major islands you can see were grouped together into clusters. Um, broadly speaking, there's maybe some like interesting traits going on, like here, for instance. It got some of my minor islands as well, although others maybe are still grouped together when maybe the t-SNE thinks they shouldn't be. But regardless, I think these results are pretty appropriate. I bet you it's separating my general major phenotypes pretty well. So I'm really curious. I was really mostly interested in separating my B cells from my T cells from my monocytes. And so it's probably done that here. And now the big question just is what do these clusters represent? So the clusters, the names of the clusters are just 1, 2, 3 through 11. I don't really know what phenotypes those represent, but I can figure it out, and I can figure it out pretty fast. So my first step to figuring this out is I can right click on this FlowSOM population I have here, right click and say make multigraph overlays, and I can make a multigraph color map. Again, multigraph color mapping will only work if the population, if it's just a single population in the chart, not an overlay. I can then choose parameters for my color map. I'll just choose everything because doesn't really hurt. And so now it's just going to make a bunch of heat maps of the different parameters on the t-SNE. And I think this is a really easy way to identify what the different expression of the different areas of the t-SNE plot is. And so I can see pretty quickly here, okay, like, let's find some of our hallmark markers. CD3, okay, the whole right side of the plot is CD3 positive. Let me zoom in a little bit just for the folks who might have small screens. Maybe even bigger. So let's focus on that CD3. The whole right-hand side you could see of the plot is CD3 positive. So that's where our CD3 cells are, And if we go back and look up in the top right-hand corner, we can see that corresponds with this cluster, and this cluster, which I can figure out the numbers for later. But let's look further at some of our specific markers. CD19 is down here. It looks like that's maybe some monocytes picking up some background signal. Um, and then let's see, CD4. Okay, so CD4 is this group here. And I think that corresponds to basically one of my big populations here in the clustering results, maybe two of them actually. So like these two populations are my CD4 cells probably. And so down here we can see CD4 is probably represented by the colors are challenging for a colorblind person sometimes, but if you click here, you can change the color. And so I do this all the time just so I can know what's what. So I'm going to just change it to a kind of a bright blue. Yeah, and we can see the color just changed. So yeah, that's number 7 is my CD4 as well as number 8 it looks like is also a CD4 subset probably. Um, yeah, so I just changed that to red and you can see its color changed. So 7 and 8 are my CD4 cells. And so if I'm interested in exploring then further about what changes are occurring for my CD4 cells between my healthy and diseased patients, I can do so. Now that I've identified and isolated my CD4 cells, I can dive in deeper. And that's what we're going to do next with Tregs. So I could identify my other subsets too this way. I could right click and rename these here in the workspace. Um, I'm not going to rename them yet just because I want to show one other way to explore the data. So I can explore them just using these simple heat maps and it works pretty well. I can also go over here, click on my FlowSOM population again, go up to not plugins, sorry. Go near plugins, go under the workspace tab to the Cluster Explorer button, and I can press this Cluster Explorer button to open the Cluster Explorer platform. In the Cluster Explorer platform, I'm going to choose my clustering analysis that I just did, which is the FlowSOM one. If I've done multiple clustering analysis, it will list all the clustering I've done, but in this case, it's just the FlowSOM, so perfect. Um, it'll show me my t-SNE map, and then I can choose any parameters I want to visualize. Again, I just want to look at basically everything except for my live dead stain. If I was looking at a subset like my CD4s, I'd want to drop anything that's not relevant to that subset. But in this case, this is fine. I will press accept and it will open up Cluster Explorer to visualize my clusters. Okay, so you can see it's opening up these different visuals. It makes a heat map down here in the bottom left, this line plot up here in the top left, a bar chart, and the t-SNE. But what's really cool about Cluster Explorer is it's all interactive. So we were just looking at that 7 population. Let's click on it again. We can actually click on it over here or over here. Um, and we can see it highlighted in all my charts. It's a kind of a hard green color to look at, especially for my colorblind eyes, but um, we can see its expression, across the different markers. And so it, of course, should have high CD3 expression, and it should have high CD4 expression. So that makes sense. The heat maps also, we can look at this. So for the heat map, we would expect, again, the labels are a little hidden, but if I just make this bigger, we can see them all. I have a lot of markers, so there's a lot of labels. But if we look at the columns, we can see CD4 is here. And yeah, it looks like 7 and 8 both have fairly high CD4 expression. Um, and then actually 10 has a little bit of it too, it looks like, but it might just be some outliers. Um, yeah, 10. So that's actually one good thing to note with FlowSOM and all of our clustering analysis. Our data is going to have some outliers in it. That's just kind of inevitable. Those outliers will usually be grouped together into 1 or 2 clusters. That's maybe what clusters 9 and 10 represent. And they might be some rare population of interest, but they might just be junk. And so, we'd have to interrogate more which the case is here. But in this case, I'm just going to ignore population 10 and just focus on, 7 and 8, which are my 2 CD4 populations. You can see they're both CD3 positive, and it looks like the difference between them is this like HLA-ADR and ICOS expression is only present in population 8. Ki67 is only present. So you can get an idea pretty fast of what the changes are between these two populations. Okay, one other neat trick we can do in the heat map here is we can add nodes as columns. Let me see if I can do it though. I can't do it right now because I haven't done what I need to do back in the workspace. I thought I did. Oh, I haven't. Um, Let me go ahead and gate once again uh the two states I have. So I did that before, but if you recall, I switched workspaces since. So I have to regate these two gates. These are my again my healthy and disease or healthy and COVID populations, the two different conditions. Um, and so this is what I'm comparing those two different groups of samples. Uh, Now that I've gated this, I can go ahead and use it back here. Oops, don't want to press that. So in this heat map, I can press this little plus sign and I can choose, whoops, sorry, it's not the plus sign. It is, I am spacing on where to add it. You can see there's a lot of options here at the very top for how to modify in ClusterExplorer some of the traits. Um, I'm totally spacing on how to add populations as columns. Oh, maybe it's a right click. Nope, that just copies it. Well, regardless, we'll just carry on with our analysis. It's not really important. It was just a little trick that there is in here. Um, let's dive into T-Rex because we only have 7 minutes left. So I have those two conditions. I have this cluster I'm interested in, cluster 7. I could group 7 and 8 together since they're both kind of CD4 populations that I'm interested in, but I didn't, and so let's just leave them separate for now. Let's take a look at cluster 7. Let's look at these CD4 cells and see what's different between my healthy and COVID conditions for cluster 7. So first of all, we can see there's a slight difference in the percentage of each of these two conditions for cluster 7. It's slightly more represented in the healthy condition. Um, so maybe, for instance, the cells are fighting and they've died a little bit. There's a little bit fewer in the COVID, although this could honestly just be some noise variance. Um, Okay, and so we want to compare these two conditions for my CD4 cells here for cluster 7. I'm just going to rename this CD4 because we know these are our CD4 T cells. Um, okay, so the way we can compare these two is we can run T-Rex to look at the two conditions on just my CD4 cells. So I can, again, click on CD4, go to my plugins, choose T-Rex, and that'll open this window here. Again, I'll want to probably choose my markers, my compensated parameters. This time, though, I want to remove any markers that aren't going to be present in my CD4 subsets. So for instance, CD4, I've already used, so I don't, this is my CD4 population, they should all have it, so I don't need to include it. CD8, similarly, none of my CD8 cells should be present in my CD4 subsets, so should remove the CD8 marker. Same with the live dead stain, same with CD3, they should all be CD3 positive, so I don't need that. CD19, none of them should be CD19 positive. I might be missing something, but that should be all right. So I just want to choose the markers that would make sense for CD4 subsets that might be present on CD4 subsets. Okay, so I have my two comparisons. So T-Rex again is going to compare two different conditions. It's going to make a UMAP and then look at the differences in the UMAP of those two conditions. It's going to use the populations underneath the population I have selected. So I selected to run T-Rex on my CD4 population. So the only subpopulations available is that COVID and healthy population that I made. And so that's what it automatically loads here. I actually probably want to switch this because the pre-treatment probably should be the healthy and the post-treatment should be the COVID. It doesn't really matter what order I put these in as long as we know which one we've assigned to the pre-treatment and which one to the post-treatment. But I just feel like healthy makes sense as a baseline. So the T-Rex clustering is going to do, it's going to make a UMAP, and so or a t-SNE. We can choose between the two options here for the comparison method. and then it's going to cluster the data based on the differences between those two dimensionally reduced spaces for the two different conditions. It's going to use k-nearest neighbors and db scan. And so I can set the k-nearest neighbors value here, and changing this will just affect the size and number of clusters that T-Rex generates, similar to changing the perplexity with t-SNE. Um, there's also this epsilon value that we can adjust for the T-Rex algorithm. The epsilon value is the distance metric that is using to assign cells to clusters. So if we increase epsilon value, we might get bigger clusters with more cells, and if we decrease it, we're going to get more small clusters. Um, T-Rex uses a lot of small clusters. That's actually one of the advantages of T-Rex and why we're doing it on this more homogeneous population, is we want to look at the small differences between my two different conditions, right? And so these aren't major subsets that we're trying to identify. These are really tiny populations that are forming because of the differences between conditions. So when I used FlowSOM earlier, I was using it to identify my major subsets, the big B cells, monocytes, CD4, etc. And now I'm really just looking at tiny differences within my CD4 population, and that's what we're trying to cluster here. Um, this bins section is where we can specify how different we want to call significant. And so again, it's comparing those two UMAP spaces, and if in a given location, 95 to 100% of the cells are from my COVID condition, it's going to put it in this first bin. So this bin represents how much of a single percentage of cells are in a location to put it in these upper bins and to put it into the clusters. So my upper bin will be 95 to 100%. And then if between 85 and 95% of the cells in a single location or in a single cluster are for COVID, it's going to put them in this second bin. And then this middle bin, which is going to be the bin that kind of represents things that aren't changing very much, if, between 16 and 84% of the cells are from one condition, it's going to put them all in the single bin because it's thinking that there's not a lot of change in that cluster, that location. Sure, there might be some difference in the percentage of cells from each of the two conditions, but it's not like this location's only being represented by one of the two conditions. And so um, that's kind of the logic here. And then at the bottom end, we have the opposite, right? So if only between 5 and 15% of my cells in a cluster are COVID, or in other words, if 85 to 95% of the cells are healthy, it's going to put them in this cluster. And then the most extreme cluster for healthy, if 0 to 5% of the cells are COVID, or if 95% of the cells are healthy, it's going to put them in this cluster. So it's just going to group basically the clusters, based on how much of only one type of the two conditions are represented in them. We can change the bounds of these bins just by deleting the number and replacing it with 7. It'll highlight red though because it's mad at me until I replace this bound as well. So the bounds need a match on either side. So if I change this to 7, I have to change this to 7. I will usually use the defaults for this when I run it. So that's what I did here. And so I'm just going to change this back. The minimum cluster size is just the minimum number of cells in each cluster. Um, I like these settings. This is where I'd go ahead and hit OK to run the algorithm. I'm not going to actually run the algorithm again, just because um, it'll take too long to calculate, or it's going to take a few minutes to calculate. I already have the results, so I'm just going to pull those up. It is also 5 p.m., so I apologize. I'm going to go a few minutes over just to show you the T-Rex results. Let's pretend I press OK to run the algorithm. It will generate these results. I'm opening a new workspace so I can open the results. I should have saved it hopefully in this T-Rex uh, workspace. Sometimes if you move workspaces, which is what I did here, it will ask you to find the derived parameter folder. When this message appears, just press find and then go to the folder that's the same name as the workspace. So in this case, it's that T-Rex folder and press choose, and it will just connect back to the results. So I ran the T-Rex analysis. You should see this is basically the same as last workspace we were looking at, except it returned the T-Rex results. When the T-Rex results return, it will do a few things. it'll make four derived parameters that we see here. I did some gating, so I'm gonna actually delete these gates really fast because I cheated. Um, if you're running T-Rex on the base sample, it'll automatically make gates, but I ran it on my FlowSOM population, so it won't automatically make the gates. So all of T-Rex will return is these four derived parameters, but if I click twice on this population, um, you will see it should have the derived parameters listed here at the bottom. And I have my clustering parameter here, and I can click on this clustering parameter. It'll have a few peaks. Um, Starkly, you'll notice the peak at the zero, that's most of my cells, because most of the areas of my plots aren't going to be changing. Most of my populations are going to be present in both my healthy and disease states. So those cells are the ones that, are not very different between my disease and healthy state, or yeah, my disease and healthy states. So they're at zero. The -1 is when T-Rex is doing its comparison, there are some so it's going to downsample to make sure there's the same number of cells from both the two populations. So in this case, one of my two populations had more cells than the other. It looks like healthy had about 10% more than COVID. So it just downsampled my healthy to evenly, take an even distribution of the cells and make it comparable in the number of cells to COVID. So that's the extra cells that it didn't use for the clustering. And then on the right here, those are our populations of interest. And so let's right click on this plot and say create gates on peaks. Um, and sometimes it doesn't refresh the visual for some reason, but if you press the refresh button, we will see it makes those gates on the peaks. I don't care about this -1 peak or the 1 peak, to be honest. So let's just delete those first two. And really, let's focus on these next three gates. Again, it's not refreshing, but if I just press the refresh button, it will. Um, I'm going to rename these. I'm just going to call it T-Rex 1, T-Rex 2, and T-Rex 3 because I don't know what these are yet, but we will work to identify them in a second. Um, and so let's look at what else T-Rex returns. So it returns these populations that I can work with. These are what I'm really interested in, but they don't mean much to me, and you can see they're pretty small. Um, So what are these populations? Well, T-Rex also returns some UMAP parameters. So if I open my graph window of my CD4 cells again and go to my UMAP, it makes really long names, but I can choose UMAP 1 and UMAP 2 here. And let's copy this to the layout. I'm just gonna uh, open my layout editor, make a new layout, and copy this into my new layout. I pressed Control L or Command L to copy it into the layout. You can right click and copy it to the layout too. Um, you'll notice the names are really, really long by default because it's being specific about what population it's referencing in the name. We can just double click on this and go to annotate and then give it a new name for the purposes of our visual. So I'm just gonna call it UMAP X and UMAP Y because the T-Rex is running UMAP, so that's what this represents. That's more manageable. Um, Okay, so I have this UMAP. This is a UMAP made by my T-Rex, though, by the T-Rex algorithm. This is the UMAP it's using to compare my two different conditions. And this visual by itself doesn't super helpful to me, but once we overlay these populations on top of the UMAP, we'll see pretty quickly where the differences lie. And really, for this case, it's just all down here. Um, you can see there's a couple of tiny little specks in here where there's some differences as well. The numbers of cells are pretty small, 33 and 18, but 2,442 of my cells are in this little group here, and that little group is unique to one of my two conditions. How do I know which one? Well, my easiest way to determine that is to go to the T-Rex output folder. So when you run any plugin, it makes a folder next to the workspace with the same name as the workspace. In this case, it's called this. Inside, there's the T-Rex output folder. You can go to output files. It makes a bunch of stuff. Um, and I actually, this top one, the healthy versus COVID T-Rex plot, I think that's the one that I want. Perfect. Yep, it is. So this is an awesome plot. You can see it has the differences plotted between my different areas. And so this is the area that's, 95% of the cells are from that COVID condition. And you can see these are the other smaller areas that it identified, but I don't really think those are that significant or interesting. It's this area that's like really only present in my COVID patients and is not present in my healthy patients. So these cells are like a unique phenotype that's forming and has unique expression of the parameters that I've used here today. And so this is like an interesting population that I want to learn more about and maybe want to identify and isolate. And so that I'm already 6 minutes over, and so I guess that is kind of an overview on how to use FlowSOM, ClusterExplorer, and T-Rex. What I would do from here, if I was doing a real analysis and I really wanted to look at this data further, is I'd probably dive into like what is this population, like um what's the expression, like what's being expressed in this population. And again, the best way to do that is one of my favorite tools, which is ClusterExplorer. To use ClusterExplorer here, what I'd actually probably do is go to ClusterExplorer, which again is under the Workspace tab, ClusterExplorer. Um, it's not going to recognize my T-Rex results as a cluster set, so I'll go down here to where it says Show Populations as New Cluster Set. I can tick this tick box, choose Configure, and choose my T-Rex populations. Um, if I can see them. Oh, it's not showing me them. Did I not choose? Did I not choose this? Oh wait, oh, sorry. I have two workspaces open. This is my own fault. This is the FlowSOM one. Sorry. This is the workspace I want to use. Cluster Explorer. Uh, show populations as new cluster set. Choose my three T-Rex populations here. Um, we can also choose healthy and disease just to see what they look like. Um, press accept. And so now, so using that use new populations as cluster set, you can choose any populations in the workspace as a thing to look at in Cluster Explorer. I can remove some of these markers potentially, um, like anything that's not going to be relevant to my CD4 subsets again. Press accept and pull up these visuals. Okay. And so the plots are going to be kind of stark because I chose to include my COVID and healthy, so it probably doesn't make sense to compare those with the two T-Rex populations, but we can just quickly see for the COVID and healthy what differences there might be between these two conditions. And actually, I see that the main difference is maybe between the expression of a couple of these markers, which aren't necessarily going to be unique to my CD4 populations. But if we want to focus on these T-Rex populations and specifically, that what was it? T-Rex 2 is the one that is the population that's being highlighted here. I'm going to change it just to make it a dark black color. That population is T-Rex 2. And so, we can go to our T-Rex 2 here and see, okay, it's expressing Ki67. It's expressing granenzyme B, I guess. Um, and so that's interesting, I guess, just to see what's driving it relative to the other populations I have selected. And then if we go back over here to this line plot, we can see just the general expression of that population again. I'm just going to click on it so we can see it. And yeah, we can see it actually has some granzyme B expression, it looks like, in Ki67, which is interesting. Um, HLA-DR. So it gives us an idea of what might be unique about these cells. Okay. So it's much past 5 p.m. now. I kind of rapidly went through this webinar. Hopefully it was helpful though. If you want to see a recording of this, because I know I was going fast and you might have to like go back through some of what we did, the recording will be available on our website in a few days. It's a long weekend actually, so it might be um, not until next week sometime, but you can get it if you go to our website and go just to flowjo.com and then go to Learn, Webinars, and then below the upcoming webinars, we have previously recorded webinars, and mine will be centered here in next week sometime. I think they'll put it on the website. I will be hanging out for a few minutes if you want to drop any questions into the chat, but it is Thursday before a 3-day weekend, so I totally understand if folks here in America want to go. If you're coming from outside of America, it is our 4th of July, so that's what I keep referring to as the 3-day weekend, our Independence Day. Um, yeah, I'll stick around for a few minutes in case folks have questions um, or comments. I can put the data set I used today in the chat in case people want to try to download it from Flow Repository. Have found Flow Repository has been hard to use recently, but maybe it'll work in your hands. Yeah, I forgot. So I just chose my three T-Rex subpopulations this time just to try and visualize them, but now it's showing me so few cells that it's hard to really understand what I'm looking at here in these 2D plots. I was just kind of curious. I can look at this a different way too. I don't use ClusterExplorer. I'm just curious where these cells are located in my original CD4 plot, my original T-SNE plot. So I can load that original T-SNE into a layout and do a quick comparison. This is all I really care about. Huh, okay, well, that's a pretty obvious population. It looks like that subset here is the one that's driving the difference between healthy and disease that we see here. Okay, well, I think I've left enough time for questions. Doesn't look like anyone has had any unless I've really been, yeah, I don't think I've missed them. Um, if you do have a question, I would hit enter now or forever hold your peace. Otherwise, thank you for joining me today for this webinar. The recording again will be on our website sometime next week. If you do have further questions, you can always email us at flowjo@bd.com, and we are happy to answer any questions via email. All right, thank you for joining me today. Have a good weekend.